So these are the last two topics we'll discuss: the Human Genome Project and the $1,000 genome. So I want to do a brief timeline of important events in genetics leading up to the Human Genome Project. 1865, Gregor Mendel, who we mentioned previously, experiments in plant hybridization, eventually leading to concepts of recessive versus dominant inheritance, other concepts like equal segregation and independent assortment. 1902, first human disease is attributed to a genetic cause. This disease was alkaptonuria, which was described by Sir Archibald Garrod, which is metabolic disease. 1944, DNA molecule which mediates heredity is shown with pneumococcus by three scientists. Many scientists are skeptical that this is that DNA molecule has anything to do with heredity. 1952, DNA again showed proven that these molecules mediate heredity. This was shown by Hershey and Chase. Now people believe that these DNA molecules are what regulate heredity in people and animals. 1953, structure of DNA discovered, double helix, by James Watson and Francis Crick. 1959, messenger RNA is shown as the intermediate between DNA and proteins. This was shown on the previous slides in that diagram that is on different slides. 1966, genetic code is cracked by researchers. The DNA and RNA code is understood for specific amino acids. 1972, recombinant DNA constructed by two scientists. This allows scientists to synthesize in the laboratory insulin and clotting factors like factor VIII used by hemophiliacs, and is a great benefit to medicine. 1977, DNA sequencing technology developed by Fred Sanger allows sequencing of small segments of simple genomes like bacteria or simple animals. As the DNA sequencing technology got more sophisticated, 1990 Human Genome Project begun as a joint venture between the U.S. Department of Energy and the National Institutes of Health. So the Human Genome Project was started in 1990, funded by Congress. The expected budget was $3 billion. It was expected to last about 15 years. A rough draft of the human genome was completed in 2000. The project was essentially completed in 2003. The project finished on budget at $3 billion, but finished 2 years early. At the beginning, the sequencing was very slow, but in about 1998, new technologies were developed which made sequencing much faster and less expensive, which allowed the project to finish ahead of schedule. This is in discussing the methods like shotgun sequencing, and also the differences of opinion between scientists such as Francis Collins and also Craig Venter, which were fodder for a lot of drama back in the 1997-1998 time period. The cost of sequencing is getting much less expensive, and this is what is going to change medicine in all of our lives. In 2007, a sequencing company called 454 sequenced James Watson's genome for $2 million. So there's also an article in the New York Times on October 6, 2008 by writer Andrew Pollack, which is titled "Dawn of Low Price Mapping Could Broaden DNA Uses." There's a company called Complete Genomics in Mountain View, California, said they will start sequencing genomes for approximately $5,000 in 2009. What they do now is they do sequencing for research organizations like big universities or other companies, where they will, say, sequence 10 genomes for $50,000, so on the average about 5,000 genomes each. And obviously they're just giving the genome sequence, they're not really doing a lot of interpretation at this price. So the $1,000 genome is coming. How will it change the world? This is an article by Luke Timmerman on May 21st, 2009. A lot of people have called the $1,000 genome the holy grail of medicine. By 2015 to 2020, sequencing a genome will likely become a standard medical test. By 2020, going to see the doctor will be much different than it is today in some locations. So what's possible? This is an example of what might happen in some places in 10 years. Family planning with your spouse. You get your genome sequenced, and your wife gets her genome sequenced, and you get a full list of your recessive and X-linked Mendelian disorders explained, and then you know what your risks are for having a child with a certain single gene disorder. You'll get a profile of your risks for most complex gene disorders, taking into account all genes, single nucleotide changes, non-protein coding RNAs, and plotting these risks versus the general population. You'll get an explanation of what diseases you're likely to get over the course of your lifetime, creating a surveillance plan and lifestyle changes to detect or reverse high-risk conditions in their early stages. A doctor may be able to tell you what drugs you are allergic to before you take them and what drugs will work best for you in the future. This will reduce the risk of having adverse drug reactions or having a bad reaction to something that you've never taken before. In some cases, treating you for diseases before you have symptoms. In some cases, in the future, detecting cancer in its early stages eventually, maybe 2025 or down the road, molecular detection and surveillance of cancer by blood testing, cancer detection at the microscopic level way before you have any symptoms. And also way before it can be detected by an MRI or a CAT scan. If you do get cancer, the doctor will take a biopsy or get a specimen of these cells and give you chemotherapy designed for your cancer. The chemotherapy drugs will work much better and have less side effects. So we're almost done here. On the next video will be our last, and we'll sum all this up of why all of this is really important.